Okay guys, let's give this a shot. So we're going to go over homeostasis in the epithelial tissues. This was the first lecture in Module 2. The other versions of it that were already done were kind of long, and I'm trying to shorten them a little bit. I'm a little bit hoarse today, so I apologize for that. But we will try to get through it anyway. Okay, so let's start. Epithelial tissue. All right. So epithelial tissue covers body surfaces and it also lines body cavities. Okay. Can do this. There we go. It only wants to write at one angle, and if I don't find that angle, it doesn't want to write. Okay. Covers body surfaces, lines body cavities. These guys are held together, which is kind of important. We obviously don't want to have like big gaps between epithelial cells because that would not allow them to communicate. It wouldn't make it durable. It'd be easy to penetrate by foreign substances. So obviously we want them to stick together. And so because of that, these are adhered to each other. And again, this makes epithelial tissues durable. And, prevents the entrance of foreign substances or pathogens, things like that. So that being the case, since it's going to prevent or help to prevent the entrance of these different substances and pathogens, it makes sense in that one of the functions of epithelial tissue is to protect. I'm going to list the others here. We've got protect, absorb, secrete, excrete, which is like to get rid of waste, and to filter. We'll see some of these functions as we go along through this particular section. Okay, so basement membrane, let's talk about that for a minute. The basement membrane is shown in this image. Oops. Anyway, it's all of this part right here, okay? The basement membrane is actually what anchors the epithelium to the variety, to its different tissues, okay? So we have a basement membrane that actually makes the epithelial tissue stick to the organs that it's part of, which is kind of important because we don't really want the epithelial tissues sliding off someplace else, right? Okay, so basement membrane then, like I said, it anchors the epithelium and epithelial tissues to the tissues it surrounds. So I'll just write it this way. Okay. And the basement membrane isn't actually made out of cells itself. It's really made out of a protein and carbohydrate mesh. And the substances that make up this mesh will probably sound familiar to you because we've got keratin, collagen, and elastin. A lot of times you'll hear these words used when it comes to like beauty care type products that claim to be able to replace this or to restore it, stuff like that. But that's where you hear it. Sorry, get out of the way. Okay. And so this basement membrane is actually secreted by the epithelial cells. So these cells are going to secrete the proteins and carbs required to build the basement membrane that will anchor it to the tissues around it. So kind of cool, I think. All right, let's talk about glands for a minute. I think I probably need to move this up for you. Make it easier to write. Okay, so let's talk about glands. Usually when you hear the word gland, you think about like a thyroid gland or an adrenal gland, and if you think about that, you realize that those are going to secrete hormones. So we're going to talk about that a little bit. Glands actually arise from epithelial cells. Here we go again. Find the right angle. Okay, they arise from these epithelial cells, and they are specialized so they can secrete specific products. Again, if you go back to the thyroid and adrenal glands are not going to secrete thyroid hormone, right? So they all have to be specially made so that they are secreting the specific product released by that gland. Okay, so let's talk about exocrine and endocrine glands. Really, we're just kind of going to do a definition, and then I'll show a couple of examples. Exocrine glands are going to excrete product outside of the body, or it's going to secrete it into like a body cavity. Okay. Okay. I always think about the EX for exocrine as being like the EX for exit. Kind of helps you remember that exocrine glands, that their products are really used outside of the body, or sometimes outside of an organ being used by another part of the body. Okay, endocrine glands, then I think about the EN of endocrine being the same as the EN in entrance, or kind of inside. Endocrine glands are going to secrete hormones or products, usually directly into the bloodstream, so it stays in the body going into the bloodstream. Okay, so a couple of examples down here. We have a sweat gland, which would be an example of like an exocrine gland, because obviously you don't sweat into your body, you're going to sweat outside of your body, right? It releases it outside. Then you've got a mammary gland, which is found, of course, in breasts, and creates milk for breastfeeding mothers. So mammary gland is also going to be like an excretory gland because it's going to come out. Pancreas is usually endocrine. It releases products that are going to be used inside the body to aid in other processes, including like digestion and things like that. Down here, there's another list of some types or some different glands. I just wanted to point out like the adrenal gland. The adrenal gland is going to be an endocrine gland because it's going to release the hormone adrenaline from the gland into the bloodstream. And again, it's largely this hormone that is responsible for the whole fight or flight response that we've talked about so much in class lately. That increases heart rate, that increases respiration, things like that. So really the trick to deciding if something is an exocrine or an endocrine gland, which will be important in physiology to say the least, it's look where the product goes and where it's used. Okay, let's talk about epithelial structures. Okay, so let's talk about the layer structures. Point out a couple of things just real quick. This is going to be a simple type of epithelial layer where we have just a single row of cells. We have the pseudostratified columnar layer. This one is more complex because you do have these long rectangular cells, but if you look down in here, we have these other cells that are interspersed throughout, so it's pseudostratified, because a real stratified layer looks like this. Kind of have these hexagon-shaped cells, and notice that they are staggered every other, kind of like if you were going to lay bricks. And so pseudostratified doesn't have this same every other, but it's pseudo. Okay. Cell shape, down here at the bottom left, we have squamous squamous cells. Say that 4 times fast. So it's kind of giving us a side view and then a top down at an angle so you can still see its 3-dimensional structure. Squamous cells are flattened. Then we've got cuboidal cells, which obviously named for a reason. They look like cubes. And columnar cells, again, they look like columns or rectangles, and so that's where they get their name. And that's how we're going to classify the types of epithelial cells and tissues that we're looking at. You look at their cell shape and their cell structure to determine what kind of tissue you're looking at. These are generally somewhat easily identified under a microscope. These different characteristics make them kind of easy to tell. Okay, so simple epithelia. This is a single layer of cells attached to a basement membrane, okay? So like this, which you can have simple squamous, simple cuboidal, or simple columnar, and those are the types we're going to look at. So simple epithelia, again, single layer of cells. Hang on, gotta find the angle for the pen. I just threw that one across the thing. Okay, I give up on the pens. Okay, so let's look at these types of simple epithelia. Move this up a little bit for us. Okay, make sure that's all in frame so you can see it. All right, simple squamous. So again, single layer, and this time we're talking about these guys, these squamous cells. They have this flattened shape. An example of where to find these in the alveoli, which are the lung air sacs. So they're in the lungs. But then within the lungs, we have these smaller structures called alveoli, where the gas exchange actually happens. They also can be found lining capillaries, which we've been talking about recently. Okay. So now let's talk about down here simple columnar cells. These are the rectangle guys. We often see these like in the digestive tract is a good example. However, you can find them in other places, like in fallopian tubes in the female reproductive system, because some of these simple columnar cells can also have something called microvilli. These are small hair-like projections that can come off some of these epithelial cells. They're usually used to help propel something, to move something. And in this case, in a fallopian tube, it's used to propel the egg out of the tube and into the uterus. Fallopian. Okay, so to propel the egg. All right, simple cuboidal. Obviously, these are our square structure guys. They're cube-shaped. These are often found in glands. And what I think is kind of cool about them is that this is one of those structure and function things where it's kind of a neat example, because their structure fits the function, which in this case is usually to secrete product, to secrete hormones. So we've got a lot of organelles inside. We've got a lot of cellular product inside, and this shape actually accommodates all of those organelles. Okay. All right. Hopefully that's all making some sense. Let's look at pseudostratified and stratified epithelia and kind of talk about a couple of their characteristics. Grab the paper. Okay. So if you remember the pseudostratified, those are the ones that look at first like they're simple, but then as you look closer you realize that there's some other cells kind of interspersed in there. Try to get this lined up a little better for you. Okay, pseudostratified. So each cell in a pseudostratified layer is attached to the basement membrane, so they're all anchored. You'll find these in the lining of the trachea as an example. Okay. And a lot of these are, well, not a lot of these, but in this location, they are ciliated. And if you remember what cilia are, as far as modes of cellular motility or movement. However, kind of like the microvilli that you can see in the fallopian tubes, when cells have cilia attached, it's also used to sweep products either toward or away from a specific location in the body. So with trachea, as you can imagine, it helps to kind of keep debris that you might inhale from going down into your lungs, and it kind of sweeps it away from there. So in the lining of the trachea, these guys are ciliated. We generally just see this type of structure in the pseudostratified layers, and also the pseudostratified layers are generally columnar. You almost never see them in the squamous or cuboidal, or made up of those of squamous or cuboidal cells. So when you see pseudostratified, it usually just says pseudostratified columnar. Unless it indicates that it's cuboidal or squamous, that's just what you should kind of assume. All right, so now let's look at the stratified epithelia. The stratified epithelia, unlike the simple and the pseudostratified types of tissues, the stratified epithelia, the cells are not all of them attached to a basement membrane. You kind of have this bottom layer that's attached to the basement membrane, but then you've got more layers of cells stacked on top of that. So the cells are kind of attached all to each other, which are attached to a bottom cell layer, which are then attached to the basement membrane. Okay. So some of the locations in the body where you're going to find a stratified epithelia includes actually a lot of the linings of a lot of the orifices in the body. So, but that, we also have skin, and then some of the orifices, we've got nose, mouth, and then we have the esophagus, and then you've got things like the anal canal, a cervix in women, the vagina in women. All of these things are made out of stratified epithelia. Okay, let me move those up so I can write. I'm like MacGyvering this whole thing. Any of you guys ever seen MacGyver? Like the original MacGyver from the 90s, and he was always just figuring things out with like chewing gum and a paper clip, and he could make a bomb, and it was amazing. Anyway, I'm not making a bomb, but I am MacGyvering this. Let's talk about the integumentary system. The integumentary system is referring to our skin, and it contains hair, nails, sweat glands, sebaceous glands, which are like oil glands, and sensory nerves. So the integumentary system is divided into layers, and we're going to go through some of the details of each layer. Obviously, because of its components, the integumentary system is extremely important, and that's probably not something I have to tell you or point out. It comes in contact with our external environment constantly. It's how we get so much of our information about life. So in order to allow us to have that information and also to prevent our bodies being invaded through the skin, again, we have these multiple layers. So those layers are the epidermis. Epi usually means on top or surface. Then the dermis. And then the subcutaneous layer. There you go, subcutaneous layer. And I know that's spelled subcutaneous, but you say it cutaneous. Let's talk about the epidermis. So the epidermis is made of stratified squamous epithelium. So this is one of those more rare examples. Oh no, I'm sorry, that's with our pseudostratified. This is common to see. We can have stratified in all the different kinds. Should be a T. Stratified. Goodness. Squamous epithelium. Okay. And if needed, I suggest you go back to the previous page that had the different cell shapes and the different tissue structures to kind of help you picture in your mind what these different layers look like. All right, so the epidermis has keratinized cells that are part of it. Keratinized. These are kind of infused throughout. You know, they have like that, I think it's called keratosis pilaris, that you can have. It's usually on your upper arms, towards the back or side, and you can get these little bumps on the back of your arms. Those are usually just kind of an abundance of keratinized cells. And there's usually some like softening treatments or something you can do that with, but we do have keratin in our skin to help with durability. Okay. They also provide us with some waterproofing, which is kind of a good thing. Imagine what would happen if we weren't waterproof when we got into a shower or a swimming pool. We probably wouldn't be getting into the shower or the swimming pool, right? It'd be like a sponge every single time. Okay, so this image is kind of showing that complex overview of all of the different parts that make up the integumentary system. The sebaceous glands, some of which are in kind of, I wonder which ones are using a sebaceous glands. These are usually sweat glands, these twisty looking guys. We've got hair follicles, we've got the different nerves that run throughout, blood vessels. These are probably sweat glands. Okay, so all kinds of different loveliness happening there. Okay, I'm going to switch pages. We're going to talk about the dermis. The dermis. Make sure it's lined up. Ish. Let's try that. All right, the dermis. So, the dermis is this next layer beneath the epidermis, and it is separated from the epidermis by another basement membrane. So we really do have these distinct and distinctly separated layers of skin. So the dermis is dense in connective tissue. It has a lot of connective tissue. Okay. It's also rich in collagen and elastic fibers. Kind of what's keeping our skin, giving us that overall shape and form that our skin has. Which is why it's often a target for different types of beauty products that are supposed to help to hide or reverse the signs of aging and give the skin its youthful form back. In addition to all of these things, the dermis also has a lot of blood vessels and sensory receptors. And those receptors are not just for touch, but they're also for pain and temperature. This is going to play into the whole homeostasis thing, guys. Okay, and sensory receptors. And again, these receptors are for touch, pain, and temperature. Okay? All right. So we're going to interrupt this here for a second to talk about our question of the day, which was seen on the front page of this, and that is, how could you cure epidermolysis bullosa? That's what this is. And so the front page showed some images. I believe it was actually one like this. What happens is you get a lot of blistering, as if you had really severe burns across your body. The skin can actually peel off as if you had been in a fire. I say you, but really this is a pretty rare genetic disorder. It affects about 1 in 50,000 people. But the symptoms are quite severe. Obviously, I've got the black and white images here, so they're probably not quite as intense as they are if you have the color version or if you're looking at this on your computer. But the only way to really deal with this is to do what the article title says, which is to regenerate the entire human epidermis. But the way this has to be done is by using transgenic stem cells. Okay, so you have to use stem cells from another source to regrow skin. And they can't be from the same individual because it's a genetic disorder, and the disorder itself is caused by the skin cells. So the skin cells that the person has have a flaw, obviously a big one, and they create these types of issues in the skin. You have to take stem cells from another source to allow them to grow completely new skin that is free from this genetic code that causes this epidermolysis bullosa. That is the long answer to that. So I have a different image. You can see this is the boy at one point. They don't have on this page another one, but it shows the boy a couple years later, this patient here, and how his skin is very well healed. This may be of the same patient, but I can't tell. The other one is just of the boy dressed in a play or something. Okay, so that's how you can cure epidermolysis bullosa, in case you wanted to know. All right, the subcutaneous layer, for some reason, does not look like it's being really addressed. Let me see if there's a spot. So they kind of, I guess we're just going to kind of add this in about the subcutaneous. Because really, technically, sorry, those are two words, it's not really skin. So this is a deep tissue layer that's made out of loose connective tissue and fat. And fat. Okay, so that's that subcutaneous layer. All right, we talked about the question of the day. Let's finish off this module, you guys. Homeostasis. There you go. Make sure it's lined up for you. Okay. All right. Homeostasis. So in homeostasis, the body maintains a constant, stable internal environment. We've talked about how the body likes stability and balance, and this is how it happens. Maintains. Okay. So what happens on a really basic level is that when a stimulus is detected by the body, whether that's coming from the integumentary system or whether it's an internal sensor, if you will, or an internal stimulus about blood sugar or something like that, the body has specific responses that are unleashed, I guess, if you will, to respond to that stimulus. Okay, so we have a response triggered. And those types of responses include kind of what we were just talking about, but I'll write them down. Sorry, let me put blood glucose or blood sugar level. pH of the blood. We talked about blood buffers, as you all loved in that last text or test section to talk about, do you need more carbonic acid or bicarbonate ion? Anyway, so pH, temperature, but there's a lot of other things too. I mean, obviously an injury, if you injure yourself, these all have their own pathways. So this circle is kind of showing how it works. Homeostasis, we have homeostasis balanced on this tip of this triangle here. Any little touch, if you will, on one side or the other will cause our nicely balanced platform to tilt to one side or the other, which our body does not like. And so in order to return back to this nice balanced state, it's going to release the response in order to bring it back to homeostasis. Okay, so here's homeostasis thrown off, which sends usually a chemical stimulus to the body, and a sensor carries that to the control center. Okay, so a sensor involves a network of nerves, and the nerves are going to carry it to the control center, which is in the brain, and different parts of the brain have different locations for different senses, actually. So the control center is going to send the response to the stimulus that we received out through the nervous system again, which is going to have an effect, whatever that is. If it's the case of blood buffering and we find out our blood is too basic, that's the stimulus, is chemically the blood recognizes as too basic. Nervous system carries the message back to the brain, which relays the message back down through the nervous system to break our carbonic acid up and release some of those hydrogen ions, right? And when that happens, homeostasis returns, it comes back to balance. Okay? Hopefully that made sense, because I can't see you to see if you're all confused. If you are, we'll talk about it. Let's go over this loop, if you will, this feedback loop as an example of blood glucose or blood sugar. So let's say that after meal, the body registers an abnormally high glucose level, okay? When it does that, we've got these glucose receptors in the pancreas that are going to activate, and their job is to trigger the release of insulin, right? Hey, blood sugar levels have gone up, we need some insulin so we can balance this out. Try to paraphrase that. Okay, these glucose sensors are going to trigger an insulin release, okay? When that happens, the insulin is going to cause a chemical reaction, essentially, that causes cells to take up more sugar. And when that happens, the liver is going to convert that excess. If you remember, for our macromolecules, one of our polysaccharides was a sugar storage molecule in humans called glycogen. And that's where this comes from, because when insulin starts to pick up more sugar, that signals the next process, which is the liver making this excess glucose into glycogen. Okay. All right, type 1 diabetes. So these are like where we've got issues, right, in this pathway. So type 1 diabetes is insufficient insulin is made. So we just don't have enough insulin. Sufficient. Put insulin production. Let me move this up a minute. Okay, so we have this insufficient insulin production in type 1 diabetes. We also have type 2 diabetes, and this is where the cells stop responding to the insulin. I know there's not really necessarily such a thing as pre-diabetes, but it can often be caught in adults later in life as being insulin resistant. That's where the receptors work, but for some reason the signal to pick up the excess sugar doesn't get sent, and so the cells don't do anything about it. Okay. Both of these types of diabetes lead to excess glucose, right, or higher blood sugar. So regardless of what type. Okay. All right. Let's talk about what happens with low glucose. Low glucose can happen if a person has been fasting for a prolonged period of time. Obviously, this would happen in cases of starvation, but it also happens if a person is purposely starving themselves, like with an eating disorder. You end up having your receptors do the opposite, where they, instead of realizing that there's too much insulin, they realize that there isn't any glucose at all. I'm sorry, I said insulin in the last one. Instead of realizing there's too much glucose, they see that there's not glucose at all. The receptors are going to detect the absence of glucose. In this case, what happens is in the liver receives a signal to break down some of its longer term glucose storage back into glycogen. So the glycogen can then be converted back to glucose so that the system can still have glucose. Let me write that and then I'll talk about that for a second. So we know that glucose is essential when it comes to some of our metabolic pathways, including cellular respiration. We've talked about how cell respiration works and that we take our 6-carbon glucose and split it down, and I'm sure you never want to hear about cellular respiration again either, but when glucose isn't present, and if we've run through our store of fats and proteins, which can also be used in the process as well, we quit making energy, which, as you know, when you quit making ATP, you die, right? So the body, to protect itself from having that happen, will dive into the liver and into those sugar storages to try to break it down, or try to break down the glycogen into usable glucose. If this condition continues, however, you can do serious and irreparable damage to the body by causing organ shutdown, tissue death, things like this on a broad scale, because obviously everything kicks into an attempt at life-saving operations only in the body. Usually a person becomes sluggish or doesn't have much energy or becomes very easily tired, and that's because the little bit of glucose that is available is being used for essential functions like brain activity and heartbeat and respiration in the lungs. So over time, if the tissues continue to be deprived of glucose, you can, like I said, have long-term organ damage and organ failure even, which is a serious side effect with eating disorders. Which, by the way, if somebody is struggling with that, come talk to me, because there's ways to help kind of navigate through that issue. But it's extremely important for the body to have this. So we've talked about low-carb diets, and obviously with diabetes, you've got to watch carb intake, but everybody else, for the most part, requires some amounts of carbohydrates, which can be quickly and easily broken down into glucose to provide energy for the body. Okay, let's talk about negative feedback. A negative feedback loop or a positive feedback loop. Sometimes you also hear talked about like in therapy as far as emotional or mental therapy, but it's a physical thing in our bodies as well. So in a negative feedback episode or stage, how to put this? I'm trying to figure out how to put this. Basically, negative feedback is the opposite of a stimulus. So a stimulus makes something happen, whereas negative feedback is going to make it stop. In fact, we kind of have, if you can still see this, the negative feedback is right here, which is making the balance stop. In this case, it's restoring the pH level to the correct place. So how do I want to put this? Opposite of a stimulus. So in this case, if this makes sense, and you kind of might have to think about it for a minute, but the stimulus, that word says stimulus, sorry. The stimulus actually is going to reduce itself or cause the reduction of itself, okay? So the stimulus triggers a reduction in the stimulus. So let's go back up here to this circle and look at our blood pH example. Blood pH is off, which is the stimulus that causes the information to be sent up to the control center, which causes the control center to send the response, like, here's what we're going to do about it. The effect then is in the form of negative feedback, meaning it's going to reduce the stimulus. It's going to correct this situation, which leads to a reduction of the stimulus, okay? In other words, it's going to bring this back to normal, and the stimulus goes away. That's what negative feedback is. Clean it up so that this stops. Okay? Positive feedback. Positive feedback is the same. The effect is the same as the stimulus. So if the stimulus takes place, the sensors carry the information back to the control center. The control center says, right, this is what we want. We need more stimulus, which in turn sends a positive feedback back down to the source that's causing the stimulus to say, keep it up, you're doing good, more stimulus. Okay? So triggers more stimulus. Sorry guys, it's hard to write down here on the bottom part. I just realized as I'm staring at my writing that I started spelling stimulus weird part of the way down the page. Oh, maybe not all the way down. Just right here. There's no O in it. I don't know what I was doing. Okay, stimulus. All right. All right, so hopefully this section made sense. Obviously, email me or come see me if you have questions, and we will go over it. All right, guys, I'll talk to you soon.